Should I start? Yep. Okay. Right. So still on topic 34. Yeah, that's fine. Yeah, I'm down muted. So still on topic 34. Yesterday we have looked at amino acids and how they can exist as and in three forms actually. Okay, so in a neutral pH, it exists as a Zwitter ion, which is called ion, but still overall it is neutral, zero charge. Okay, and then it depends on the pH value of the solution. They can exist as a positive ion or a negative ion. And then we also looked at how amino acids can be combined together in a condensation reaction, or this can also be called an addition-elimination reaction, because as we combine them, we lose or we form water. And then we also learned that the peptide bonds can be hydrolyzed the same way like the amide bonds, because peptide bonds and amide bonds are basically the same. The only difference is that peptide bonds is the name given specifically for when you put amino acids together in a long chain. Okay, so today, the last part, we're going to look at electrophoresis. The learning objective is to interpret and predict the results of electrophoresis on mixtures of amino acids and dipeptides at varying pHs. The assembling of the apparatus will not be tested. Okay, so this is how a setup of an electrophoresis or electrophoresis looks like. Basically, you have your slide. Usually it's made of glass, okay, your glass side. This is where your sample is, and the sample is, it contains a mixture of amino acids. Then one side of the setup is connected to the positive terminal. Okay, so there's a power supply here, there's a current, there's a potential difference. And then the other side of the plate is connected to the negative electrode. So you have a cathode anode here, right? Everything else, you don't have to worry about how it works or how to set it up. You just have to know that once the current is supplied, or once there's a potential difference, the mixture of amino acids will move at different rate and also at different direction depending on their overall charge. Just like chromatography, I think you're all familiar with chromatography sample where you have one dot starting at the bottom, and then the solvent will carry out, will move the sample along, and then some colors or some, in your case it will be color, but for chromatography later on we will see some molecules will move further up, some molecules will only travel a short distance. Okay, so this is a separation technique. The same way, electrophoresis is a separation technique if you want to separate the individual amino acids from its mixture. Okay, provided that they have different size, different charge, and they move at different speed along the what we call the electroferrogram. Electroferrogram is just like a chromatogram. After you carry out the chromatography, you see there's the paper of the chromatography is a chromatogram. So in this case, it's called an electroferrogram. Okay, so electrophoresis is used in biochemical analysis to separate, to identify and purify proteins. It can be used with amino acids and peptides obtained when a protein is hydrolyzed. So you need to break down the proteins or the peptides, polypeptides first into individual amino acids, then only the amino acids can be separated. Based on the different rate or the different speed that they move under the potential difference or under the given current. Okay, the technique is based on separating ions placed in an electric field. The sample to be analyzed is placed on an absorbent paper or on a gel supported on a glass plate. So this plate is made up of like paper, just like your chromatography paper. Then you will have a buffer solution to carry the ions along. The rate at which the ions move towards the oppositely charged electrode depends on the size and the charge on the ions. Okay, of course, ions that are positively charged or amino acids that are positively charged will be attracted to the negative electrode or the negative terminal. And the opposite is also true. And the size will also affect the speed or the rate at which these amino acid ions move towards the electrode. Okay, so larger ions will move more slowly through the electroferrogram. Highly charged ions will move more quickly. The reason for this is the bigger the charge is, the stronger the attraction it is to the electrode. Right? So you can just imagine, since this plate is connected to the positive terminal of the current, the power supply, so that means this plate is like acting as your positive terminal, and this is acting as your negative terminal. So your amino acids, anything that is negatively charged, will go in the left direction, okay? And your positively charged ion will go on in the right direction, okay? And then again, it depends on the size of the charge and also the mass of the molecule. The heavier they are, the slower they move. So those are the two factors that you need to remember, okay? So size or mass and the charge, okay? All right, so the ions are separated as the electric field is applied. A series of lines or bands are obtained on the paper or gel once a chemical is applied. So amino acids are colorless. Okay, so ninhydrin or non-hydrin is like a coloring agent. So it's like a spray that you would spray on the paper so that you can see the spots or the chemicals, okay? Because amino acids are not colored. Unlike chromatography, where we use marker pens, those spots are colored, right? We're separating the colors. But for amino acids, they are not. So we have to spray using a coloring agent just to turn the colorless amino acid into something that is visible. Or you could use it under ultraviolet UV light. Okay, sometimes they appear to be a certain color under the UV light. Okay, so the series of bands is called an electroferrogram. Okay, the charge on the amino acid ions depends on the pH of the solution. Therefore, the movement of ions to the electrodes will be affected by pH. Now, just now I was mentioning about amino acids in the form of their ions. You must also remember that at certain pH, these ions, these amino acids will develop a certain charge, okay? Like if it's a pH 7, it would be neutral. But if it's high pH or low pH, we learned that at low pH, it will be positively charged ion. At high pH, it will be negatively charged ion. However, we also haven't taken into account of the R group. Sometimes the R group also contains an acidic group, COOH, or it contains an amine group, NH3. Okay? So when we look at the example, so here's example 13.1. We're going to look at carrying out electrophoresis on this 3 different amino acids mixture, okay? So what I mean by we haven't taken into account of the R group. If we look at amino acid A, okay, so we know that at neutral pH, okay, the COOH becomes CO minus, and then the NH2 becomes NH3 plus, all right? But the R group, this is called the R group, also contains an NH2 group. Okay, this NH2 group has been protonated. It has accept a H plus. So if you look at the total number of charge in this molecule, you've got +1, +2, -1. Overall, you have a +1 charge. Okay, so here +2 plus -1 is +1. So overall, this amino acid has a charge of +1. Amino acid B, which looks like a glycine to me, the R group is H. So I'll just label the R group. But you're not expected to know about the R group anyway, so you don't have to worry about that. You can see there is only one amine group and one acid group, and the charges cancel each other out. Okay, so this is a neutral molecule. It's not an ion. And then last one, amino acid C. The R group this time contains a COOH group which has been deprotonated. It has lost its H plus. So you've got 2 minus plus 1. So the charge is -1. Okay? So you can write that down in the box. Amino acid A has an overall positive charge, +1 in a certain buffer solution. Amino acid B is neutral, and amino acid C has an overall negatively charged, so -1. Okay, so what happens if we mix all this amino acid A, B, and C, and then we are asked to distinguish, okay, identify which is which one is A, which one is B, which one is C. Thankfully, they all have different charges, so they will all move in different direction. Before we look at the electrophoregram, let's just talk about amino acid A. Will they be attracted to the positive or the negative terminal of the setup? Negative terminal. Okay, so you would imagine anything that moves towards the negative terminal will be amino acid A. What about amino acid B? Will it go to the positive, negative, or it stays? It stays. It is not attracted to either one of the electrode. And lastly, amino acid with a charge of -1 will be attracted to the positive terminal. Okay, and that is how we see in the electrophoregram. This is an example of an electron ferrogram. Side view will look like this. Okay, this is the positive terminal. You started at the center. That's your starting point for all the 3 amino acids. You can see that amino acid B is neutral, so it does not move to any of the electrode. And then amino acid C, which is negatively charged, it has a charge of -1. It moves towards the positive terminal. And amino acid A with a charge of +1 moves towards the negative electrode or the negative terminal. Okay, and that is how you are able to tell, say, if you did not know this, okay, you don't know which is which, then you should be able to tell that, well, this would be negative ion, this would be neutral because it doesn't move from its starting material, and this would be a positive ion. Okay? Right, and that's the simplest electroferrogram that you can see. That's because they all have different charges. Sometimes you will see that you may have two ions that are positively charged, okay? They may have the same charge, but their masses are different, right? That's still easier to see because the heavier one will move slower. But there's also a case where they both have different mass and different charge, okay? So that's when you have to look at the charge density. All right. We'll look at examples for that. I don't have an example now in the notes. The last one is just to explain how the pH of the solution can actually affect the charge, the overall charge on the amino acids, and so it will affect their movement along the electrophoregram. Okay? And that is the end of this topic, just a short one. Nina, you can stop the recording. Next, since you do not have your exercise 33, I will just distribute